Hello, everybody. My name is Abby,
And my name is Ellie.
and we will be teaching you about the creation and usage of
Bacterial ghosts. To start things off, we would love to give a special thanks to the NSF grant program for high school students. This program allows students to meet scientists in Iowa and learn about their studies. With this knowledge and our big brains, we turn complex studies into simplified versions, easier to understand for the general audience.
The bacterial ghost study is being conducted by the current University of Iowa graduate student, Christopher Vidmar. Chris, what a legend, has been conducting research throughout the past 5 years with his team of other scientists to discover the best way to create a safe and successful bacterial ghost. Dr. Jennifer Fiegel overviews the study. A little background on Chris. He completed his undergrad at St. John's University with a major in chemistry, and then transferred to the University of Minnesota for his master's. In 2020, Chris came to Iowa. After his third year, he received an internship at Boston Scientific. The goal of his current research is to target a specific drug area instead of having drugs administered throughout the whole body.
Before getting into our exciting topic, bacterial ghost, it is crucial to understand how the body works. Our body contains many receptors that allow for the body to respond to the environment. When you're administered a drug or a vaccine, it doesn't go to a specific area. It is spread throughout the body. This can result in sickness or weakness, as seen in a flu vaccine when receiving one.
Going into a little more detail, our body has receptors that accept these influxes of administered medication. They are called G-protein couple receptors, or, for short, GPCRs, which is what we'll be referring to them as for the rest of the presentation. Nearly 30% of FDA drugs target these GPCRs, which, meaning this bacterial ghost thing is going to be pretty dang good. There are hundreds of these receptors throughout our body. A wide variety of interactions can occur here, but for the sake of this study, we will only focus on one. This interaction is called an adherence reaction. Adherence reactions are when a bacteria or any type of foreign body attaches to these receptors found on multiple different tissues, areas, and organisms. When they attach, it's called adherence. The project goal is to find the bacterial receptor adherence interactions that help bacteria localize in specific regions instead of an overall body reaction.
Now, going to the fun part, the specifics of bacterial ghosts. The goal of these bacterial ghosts is to interact with the GPCRs and specifically attach to them, as GPCRs are found more highly in diseased areas of the body. When observing these bacteria structures, we want to maintain this shape and look with no insides. In short, bacterial ghosts are a hollowed out bacterial shell. No way, almost like that's where they get their name. The outer shell stays intact, but the insides are removed and replaced with a drug for that certain diseased specification. In the current study, which is being conducted, the hollowed shell of 3 different types of E. coli are being used at this time. Hopefully, when the ghosts are administered into different cells, we can observe GPCRs latching on to our bacterial ghosts. After these cells are attached, they release the medication into that specific area in the body. When allowed into a system, we want to deliver the drug to the diseased area, into the GPCRs that are targeted.
Now into the meat of the project, testing the bacterial ghosts. To test them, they use 3 types of E. coli strain like stated earlier. The specific human cells that are targeted are synthetic human kidney cells. It is not yet tested in humans. The amount of ghost concentration is also varied amongst the results. When tested, the behavior also varied among the strands. The process of the bacterial ghost hollowing is as followed.
Before we go into specifics on how exactly this process works, we need to talk about the centrifuge. Now, the centrifuge is a machine that they use to actually remove the internal components of the bacterial ghosts. Over here, I have a little example of it. Now, this is not exactly what Chris and his team use, but it's just a rough kind of on-the-market centrifuge. It's typical in a lab setting. They have a little bit larger one that they recently purchased, so now their process can go a little bit smoother. Internal components to this will look similar to this, or that. Now, these can vary among what type of machine they have, what size, every part of that will matter. But for this aspect, we'll just use this. This is kind of a rough guesstimate to what they use. Now, the internal component will be rapidly rotated. And to show this, we have a little example. And now, as you see, we have a bacterial ghost. Now, this whole process is done through the rapid spinning, where an incision is made on the actual E. coli cell. So then we can have that pressurization occur, the inside's removed, and then the outer shell staying intact. That pressure will cause that bacterial ghost to stay intact, and then remove the unimportant stuff, that middle. This will all be also done with chemicals, which will be seen in the next step. Back to you.
Buckle up, guys. This is a long one. 72-hour static culture. Then centrifuge 10-minute wash with 0.5 NaCl. Then treat with NaOH, CaCO3, and SDS. Then, you guessed it, centrifuge and wash. Treat with H2O2. Oh, centrifuge and wash. Treat with ethanol. Oh, one more. Centrifuge and wash. If the wash process is successful, then you will be able to see purple dots on the stain slide that is stained with crystal violet. The number of ghost bacteria can be increased, but there's a limit or max to how many they can do at a time. When tested in a modified version of a kidney cell, there was a positive result. There was a cellular response to a bacterial cell, signifying that the testing would work. The 3 tests that they completed were as follows. 1 ghost to 1 kidney cell created 28 hits of the GPCRs and the bacterial ghosts. And the next, they did 10 ghosts to 1 kidney cell, signifying 25 hits. The next was 100 ghosts to 1 kidney cell, which only resulted in 17 hits. There is a max and there is a limit to how many bacterial ghosts you can do at a time. And from the looks of it, it's a lower number. In the actual experimentation of the receptors accepting these bacterial ghosts, 2 plates were used. In the first one, a white plate was used in a well-established experiment to identify if the bacterial ghosts interact with the GPCR to activate a response. The exact response is unclear and needs more studying to investigate this interaction. On the other hand, black plates were used to see if we could adapt the well-established experiment for something more aligned with our project goals. We were specifically looking for bacterial ghost GPCR interactions that cause bacterial ghost adherence. In other words, could we establish methods to find GPCR bacterial ghosts to adhere to? There is a sweet spot for how many ghosts would be needed to ensure medication is as beneficial as it can be. Noted, this process is completely new. We are still developing, still testing, still processing, and with this processing, with this testing, we have not yet tested it in humans. But, on the other hand, it has been very successful in animal testing.
There we have it. Bacterial ghost. It would allow us with so many capabilities into the human body. Starting off, bacterial ghost would be allowed to insert certain medication directly into a diseased area with drugs or vaccines. In this way, medication can be implemented in so many beneficial medicational uses to ensure we target only the disease area instead of the whole body taking in possibly harmful medications.
Overall, this system is very beneficial, and all of the outcomes are looking very well. But you still have to consider the body is a very complex system, and this medication still needs to be tested. We would like to give a huge thanks to Chris Vidmar and his team for allowing us to learn more about this amazing lab. What he and his team are doing is groundbreaking and is the very future of medicine.
And we would love to give a special thanks to the NSF grant program. Without you, we would have never had this extraordinary opportunity to see the things we did and share our information with the world.
From Abby
and Ellie,
we hope you enjoyed and learned a little more about bacterial ghosts.
Hope they don't scare you on the way out.